right, on to the inner layer or retina. So the retina has a thin outer pigmented layer just deep to the choroid layer that absorbs light. It has an inner neural layer that contains photoreceptors, support cells, and neurons that perform preliminary processing of visual information. This layer also contains blood vessels. Yes, there are blood vessels on the inner layer of the neural layer. And if you look in the picture on the left and you look at the bottom where there's a little arrow pointing light, you see that there's a capillary right there. And I was always amazed by this. I was like, well, doesn't the capillary get in the way of the light? But apparently, no. They're thin enough and tiny enough that they don't. And evolution has taken care of all that. So great, our eyes do work. All right, the neural layer has several layers. The outermost layer contains around 130 million photoreceptors. And the photoreceptors are rods and cones. So just to keep us oriented on our layers, when I say the outermost layer, this would be the layer that was the nearest to the choroid layer. Peeking back for a second at our choroid, the vascular layer, we see it underneath the retinal layer. So when we talk about the outermost layer, we're talking about the layer that is closest to the choroid. And it's that layer that contains 130 million photoreceptors. So again, a little surprising to me because before you get to the photoreceptors, the little rods and the little cones, there is not only a capillary, but other tissue and layers of cells before the light quite gets to the rods and the cones. So interesting. It's just the way it is. Very interesting. The pigmented layer is a very small layer. If you look at the little picture on the right, it's showing the choroid, pretty thick. Then the pigmented layer of the retina is just one single layer of cells. And then all of the rest is the neural layer, okay? There's the innermost layer, the rods and the cones, and then there are different cells in the layers that are deep to the neural layer. No, not the neural layer. How should I say? The rods and the cones. It's all part of the neural layer. Did you get that? So I was saying choroid, pigmented layer, rods and cones, and then bipolar cells, ganglion cells, deep to the rods and the cones, and ultimately beyond that, we even have little capillaries. Okay. So around 125 million rods are found in a broad band around the periphery of the retina. They are stimulated by very little light. The stimulation of rods creates black and white vision. And apparently they did a research study, where they were shooting one single photon and seeing if people could perceive a single photon. And they were able to, at least some of the time, even though they were somewhat uncertain about their answer. Like, did you see anything? I don't know, like that a little bit. But yes, I think so. And they were right. They could see it. One photon. My goodness. It's crazy. Evolution. Okay. Around 6 million cones are found towards the center of the retina in an area called the macula. And look on the picture here and be certain that you see that the macula is surrounded by the dotted line. Inside the dotted line is the macula. And then the highest concentration of cones is in the center of the macula in an area called the fovea. And note that they're pointing at that more pigmented spot in the middle of the macula. When we look directly at an object, its image falls right on the fovea, the place with the most cones. Cones require more intense light than rods to be stimulated. Okay, so I just, was saying that just one single photon was stimulating a rod, but more intense light is needed to stimulate a cone. So, and you'll notice, like, if you've ever been out in the moonlight, for example, and you look around at the world, the colors are gone. It's like they say by the light of the silvery moon, because you're seeing in black and white, like gray, and black, silvery world. Okay. There are 3 different types of cones: blue, green, and red. They are named for the wavelength of light to which they are most sensitive. The stimulation of cones creates color vision. So if you look down here, it's a great little chart, and it's showing you that the blue cones, for example, which have a certain wavelength, detected best by the blue cones, but there's it's not just at the tippy top of the blue cone that the retina is stimulated. The retina can be stimulated by wavelengths of light that are slightly lower and quite a bit higher. The blue cones have a range, in other words, that goes from maybe 400 to 550 nanometers. And you can do that for each of the sensory receptors, the blue cones, the rods, the red cones, green cones and red cones, and kind of appreciate that the chart is showing us the wavelength that the receptor is most sensitive to, and there is a peak area of stimulation or of retinal response. But look at red, it goes all the way from 500 nanometers to 700 nanometers of wavelengths of light can stimulate it. So it's interesting. Okay, so rods and cones synapse with bipolar cells, and bipolar cells synapse with ganglionic cells. And then ganglionic cells send action potentials to the brain. There are also horizontal and amacrine cells that modify communication between the photoreceptors, the bipolar cells, and the ganglionic neuron. And you actually don't have to learn their names or anything. They're just in there, and I just put them in because it's only fair. So going from closer to the choroid and down, the horizontal cells you see up there, they look like an upside down purple octopus. And then the amacrine cells, they are in the layer that is deep to the outer layer, and they look like brown octopuses. Right? The axons of the ganglionic cells bundle together to form the optic nerve, and the optic nerve leaves the posterior of the eye at the optic disc. A central artery and vein run right through the center of the optic nerve. So here's our optic disc, and you can see that all the branches are collecting themselves like water that's going to go down like a sink or something. The vessels are all collecting, and they're going to exit the retina and go into the optic nerve. Okay, so there's the optic disc, there's the optic nerve, and the central artery and vein. There are no photoreceptors at the optic disc. Therefore, this area does not detect light and is a blind spot in our vision. However, the eyes normally make small involuntary movements that fill in the blind spot, and we are completely unaware that it's there, okay? And in case you were wondering, what is this picture anyway? This picture is called a fundoscopic picture. This means that you've taken a light with like a magnifying lens on it that is used in clinics, and you're shining the light through the pupil and looking at the interior of the eye. The little handheld device with the light and the magnifying lens is called a fundoscope, and this is the fundus of the eye and that you're looking at. And the process of looking with your fundoscope is called fundoscopy of the eye. And when you do clinical training, you'll learn how to do this. And you can actually look right into the eye and see all of these things with no special prep or anything. You'll see the macula, the fovea, the optic disc, the optic nerves, other capillaries on the surface of the inner neural layer of the retina. And of course, there's all kinds of pathologies that can be detected, which is why you're even looking in there, okay? Why do you care, right? So, because it can be, you can look at stuff and actually see certain disease processes, let's say hypertension, diabetes, clots in the optic vein that are causing loss of vision, all different things. On to cavities and chambers. The lens separates the eye into an anterior cavity and a posterior cavity. The iris separates the anterior cavity into an anterior chamber and a posterior chamber. I would recommend thinking a little bit about these two anatomical words that are being used here. So the lens separates the eye into an anterior cavity and a posterior cavity. The iris separates just the anterior cavity into an anterior chamber and a posterior chamber. So the chamber's in the front of the eye, okay? The chambers are in the front of the eye. The cavities are in the whole eye, because there's an anterior one and posterior one. So because they both start with a C, and a chamber sounds a lot like a cavity, sort of in our minds, you need a little attention to that so you don't get mixed up. And here's our picture showing that the aqueous humor is purplish in the anterior cavity, and then our posterior cavity is full of vitreous humor, and it's got a sort of pale orange color. And there's that lens separating the anterior and the posterior cavity. And then there's our iris separating the anterior and posterior chambers that are colored purple. And this is a super close-up. The eye is filled with two humors or fluids. The anterior cavity, both the anterior and posterior chambers, are filled with aqueous humor. And remember, the aqueous humor is made by the ciliary processes of the ciliary body. Aqueous humor is similar to cerebrospinal fluid. It transports nutrients to and waste from the different structures in the anterior cavity, both the anterior chamber and the posterior chamber. The anterior chamber and posterior chamber are connected by the pupil and share aqueous humor. The aqueous humor is made by the ciliary processes of the ciliary body, and then it flows through the pupil into the anterior chamber, and the aqueous humor drains from the anterior cavity through the scleral venous sinus, which used to be called the canal of Schlemm in the anterior chamber. So, scleral venous sinus, it's descriptive. It's good. I just missed the canal of Schlemm. What can we do? All right. But the important thing is that you learn it, and that you learn that it's draining the aqueous humor from the anterior cavity in general. The canal of Schlemm, or the scleral venous sinus, is in the anterior chamber area, way off on the side of the lateral part of the pupil. So way over there. The posterior cavity contains vitreous humor, a gel-like substance that helps the eyeball keep its shape and presses the retina flat against the posterior eyeball. The lens is composed of a dense fibrous capsule, an epithelium, and inner layers of lens fibers. So again, the lens is a very clear structure, and I was surprised to learn that it has a complicated structure overall with the lens epithelium, lens fibers, and the lens capsule on the outside, the lens epithelium, the picture is very clear, and then the lens fibers deep to the lens capsule and the lens epithelium. Okay, vision, and we're done. So, vision is a special sense. The visual cortex is larger than the cortical area for any other special sense. Each rod and cone monitors a specific visual receptive field. A visual image results from sensory input from the entire receptor population. The optic nerves, bundles of the axons of ganglionic neurons from the retina, leave the posterior of the eye. Axons from the medial portions of the retina cross at the optic chiasm, or crossing, and then these axons carry input from the lateral visual field. So we look at our picture and we look at what light rays are hitting the medial retina. We see that the light rays from the lateral visual field, let me put lateral left visual field, and the light rays that are hitting the medial retina are also from the lateral right visual field. And then the light's coming in, and they're coding one little axon here, blue, and it's going to go in. And then it's going to go from the left into the right optic chiasm and vice versa. The axons of the ganglionic neurons travel to the thalamus in the optic tracts, which are beyond the optic chiasm. They synapse on neurons in the thalamus. Visual input from the left half of each retina goes to the left thalamus, and from the right half of each retina goes to the right thalamus. So we already tracked what happened to light that was coming from the left lateral field. If it went to the medial eyeball, the axon went in, it crossed at the chiasm and went to the right side of the brain. But we have another part of the visual field. So we'll take our left eye again, and now we have the medial left field of vision, and it's going to come in, it's going to hit the lateral part of the retina, and then it will send stimulation down the axons, and it will stay on the side that it originated from. So it's interesting. Make sure you look at this picture carefully. The thalamus relays visual input to the occipital visual cortex. The visual cortex contains a map of the field of vision, and visual input is delivered to the appropriate area in the map. Visual integration and association areas process the visual input and create a composite picture of the world by the miracle of evolution. Okay? So make sure you know optic nerve, optic chiasm, optic tract, which is actually bringing information from the medial left visual field and the lateral right visual field. They've color coded it for you in blue and blue so that you see there's information from the left lateral visual field and the right medial visual field going into the right side of the brain. All right, guys, one last slide. So some visual input bypasses the thalamus and goes to the superior colliculi, where it stimulates involuntary motor responses. Some visual input goes to parasympathetic nuclei in the midbrain that send motor commands to the sphincter pupillae muscles to constrict in bright light. And some visual input goes to the hypothalamus and the pineal gland. This input helps establish a day and night and wake and sleep cycle. So to count up places that the visual information goes, it goes to the visual cortex, it goes to the superior colliculi, it goes to the midbrain, it goes to the hypothalamus, and it goes to the pineal gland. So we didn't expect that when we started learning about vision, right? That the stimulation of our light receptors, photoreceptors in our eye would talk to so many parts of our brain. All right, very good. There is a whole part in green below that is about the cow eye dissection that we're going to do. And it tells you to look for certain structures, and then it carries you through the dissection. And you can pre-read that or definitely read it in class, and we'll go through it together and make sure that everybody has seen every single structure on this list. And it'll be really fun. And actually, it's really fast. There's not much to the cow eye. So more dissecting in your future. All right. Thank you, and see you soon.